Hello, everyone, and welcome to today's session. And today we'll be discussing the theory for the chapter Cell, the Unit of Life. Okay, so today in this session, we'll be discussing the NCERT section 8.4. Okay, and within this section, we'll be discussing 8.4.1 and 8.4.2. Okay, this is the part 2 of the lecture. Before starting the lecture, I just want to give you information that the images, text, and other materials used in our presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. Okay, so what are the objectives of today's session? So we will understand the structure and function of the cell envelope. Okay, so we'll talk about the cell wall, cell membrane, as well as the capsule. Clear? Then we will identify various modifications of the cell envelope. Okay, so there are modifications of the cell envelope. We'll talk about these modifications. Then we'll also describe the structure and role of the prokaryotic ribosomes in protein synthesis. Okay, so we all know that prokaryotes and eukaryotes, they have different ribosomes. So we will understand the role of prokaryotic ribosomes in protein synthesis. Here we'll be talking about polysomes. Okay, when multiple ribosomes attach to the messenger RNA. Then we will recognize different types of inclusion bodies. Okay, and what is their significance in bacterial cells? Okay, so in today's session, we'll be talking about the prokaryotic cell, which is NCERT section 8.4. Okay, within this topic, we'll be discussing cell envelope and modifications of the cell envelope. Okay, then we'll be talking about the ribosomes and inclusion bodies as well. Then next, we will summarize this entire lecture. Okay, we'll discuss some NCERT based questions related to these topics. There will be some keynotes that will help you memorize these topics, and there will be some interesting information for you as well. Okay, and then there will be some interesting questions for you that you have to critically analyze, you have to critically answer them. Okay, so these questions will enhance your critical thinking ability. So let's talk about the prokaryotic cell. Okay, so prokaryotes, they are represented by bacteria. Bacteria is a prokaryotic cell. Prokaryotes are included under kingdom Monera. Blue-green algae, this is also a prokaryotic cell. Blue-green algae is also known as cyanobacteria. Okay, so blue-green algae is a eubacteria. It is a true bacteria. Bacteria can be categorized into two groups, archaebacteria and eubacteria. Then you have the mycoplasma. Mycoplasma is also a prokaryote. Okay, now what is the unique thing about mycoplasma? Okay, so mycoplasma is the smallest living cell and it lacks the cell wall. It can survive without oxygen also. It can cause disease also. Okay, now next one is PPLO. What is the full form of PPLO? Pneumonia-like organism. So these are all prokaryotic cells. Now, what was the feature of prokaryotic cells? Prokaryotic cells, they lack a true nucleus. Instead of nucleus, they have the nucleoid, which is a region where the genetic material is present. So genetic material floats. Okay, because there is no nucleus. And this region where the genetic material is present, this is known as nucleoid. Then they lack membrane-bound organelles. They have 70s ribosomes. Prokaryotic cells are smaller as compared to eukaryotic cell. Now let's talk about the size of the prokaryotic cell and how prokaryotic cell divide, or how do they reproduce, you can say. By reproduction, what happens? Okay, reproduction is not just producing progenies or offerings. Okay, so when a cell divides into two cells, this is also a kind of reproduction. There is increase in the number of cells, you can say. One cell is divided into two, like binary fission, it is reproduction. One cell divides into two, this is also reproduction. Okay, so prokaryotic cells, they are generally smaller than the eukaryotic cells. So they are 10 to 100 times smaller than the eukaryotic cells. Now, prokaryotic cell, they reproduce more rapidly than the eukaryotic cells. Okay, so we all know that bacteria, it can divide rapidly. And human cell, what is the lifespan of a human cell? Or you can say the human cell divides into two in how many hours? Okay, human cell divides in 24 hours. Okay, bacteria like E. coli, their doubling time is 20 minutes. Okay, so bacteria or E. coli, that is a bacteria, that is a prokaryote. They can reproduce very quickly. Okay, eukaryotic cells, they divide slowly. Like human cells, they divide in 24 hours. Okay, so this property of prokaryotic cells, that they can divide very rapidly, this is also used for synthesizing some biological products in large quantities. So, in bioreactors, these bacteria are grown in a culture media. Okay, and then these bacteria, they divide and they divide and then they re-divide, and billions of copies are made, and the biological product, this is produced in large quantities. So through genetic engineering, we can modify the genetic makeup of the prokaryotes. Okay, we can modify the, you can say, DNA of the bacteria. We can insert human gene into the bacteria as well. Okay, so as the bacteria will divide, more copies of the gene, the human gene, will be produced, or any other gene will be produced, and you will get product in large quantity. Okay, clear? So prokaryotic cell reproduce more rapidly than the eukaryotic cell. Prokaryotic cell, this is very simple. It has a simple organization. Eukaryotic cell, they are little bit complex. Here you can see in this image is showing you, this is a diagram showing comparison of eukaryotic cell with other organisms. So you can see this is a typical eukaryotic cell shown you on the left side. Okay, the size is 10 to 20 micrometer. Okay, this eukaryotic cell, this is large, you can see. This consists of nucleus. Okay, within the nucleus, the genetic material is present, which is DNA. Okay, then here the organelles are also present. Membrane-bound organelles are present. So you can see mitochondria, Golgi bodies, endoplasmic reticulum. Okay, ribosomes you can see. Now these images on the right hand side, this is showing you what? So this is the typical bacteria you can see. Okay, size is 1 to 2 micrometer. Then PPLO, what is the size? About 1.1 micrometer. Then you have viruses. What is the size of virus? 0.02 to 0.2 micrometer. So viruses are even smaller than bacteria. Okay, so these viruses, they can pass through the bacteria, you can say, proof filter also. Okay, So there was some filters which can block bacteria, but these filters were not able to block the virus. So viruses, they are even smaller than the bacteria. Viruses can infect bacteria also. Okay. Now, what is the name of viruses which can infect bacteria? So this is known as bacteriophage. Now next, morphological characteristics of the prokaryotic cell. Now, they show significant variation in shape and size. Okay, like bacteria also, they can be grouped into various categories based on their size. Okay, now what are the different groups? First one is rod shaped. Okay, bacteria can be like rod like. Example is bacillus. Then they can be spherical. Example is coccus. Bacteria can be comma shaped like Vibrio cholerae. And then spiral. Bacteria can be of spiral shape also like spirillum. Clear? So here you can see this is image showing you different kinds of bacteria. Bacteria has been divided into different categories based on their shape. So you can see here, this is this spherical. Okay, bacteria. Clear? Example is cocci. So you can see these are spherical bacteria. Clear? Examples are diplococci, then staphylococci. Okay. Then you can see these are the rod-shaped bacteria. Example is bacilli. So you can see these are rod-shaped bacteria. Clear? Example is Bacillus anthracis. Salmonella typhi. This is also a rod-shaped bacteria. Okay. Clostridium botulinum. This is also a rod-shaped bacteria. Clear? Which causes, you can say, your food toxicity. Then spiral bacteria. Okay. Example is Spirilla. And this is Vibrio cholera. Vibrio cholera is a comma-shaped bacteria. Okay. Here also you can see these are bacteria of different shapes. This is cocci, which is spherical. Bacilli, which is rod-shaped. Spirilla, which is spiral shaped. And then you have the Vibrio cholera, which is comma-shaped. Okay. Clear? So these are the bacteria. Okay. They are cellular structures. Viruses, they are acellular structures. Virus is a link between living and non-living. So whenever they are in the host, they can replicate because they have the DNA, which is protected by the protein code, which is known as the capsid. Okay. So when they enter the host cell, they can divide by using the host machinery. Clear? So they don't have the machinery to divide, they use the host machinery to divide. So this is how they can grow, they can divide, they use the host machinery. Okay, bacteria can divide by itself. Okay, even the bacteria consists of a plasmid. Plasmid can also self-replicate. Now let's talk about the key features of the prokaryotic cell. Okay, obviously we all know key features of prokaryotic cell. First one is they don't have a well-defined nucleus, means they lack a true nucleus. Now next, genetic material is naked, means not enveloped by a nuclear membrane. So genetic material is floating in a region known as the nucleoid. Okay, genetic material is DNA. Okay, genomic DNA is present in a region known as nucleoid. Okay, now apart from this genomic DNA, many bacteria have small circular double-stranded DNA outside the genomic DNA, which is known as plasmids. Okay, so bacteria also consist of DNA apart from the genomic DNA. which is known as plasmid. So plasmid is extra chromosomal DNA. Okay, it is not chromosomal DNA, it is extra chromosomal DNA. This is small and this is circular. Okay, it is not linear, it is circular DNA. Okay, and it is double stranded. Clear? So these plasmid, they are also used as a vector in biotechnology. Okay, these plasmid also consist of genes which help in the selection. So they consist of selectable markers such as antibiotic resistance. Clear? These plasmid, they are used as a vector. What do you mean by they are used as a vector? Means any gene, any DNA fragment can be inserted into these plasmid, or they can be inserted into these vectors. And then this plasmid in which the foreign DNA has been inserted, this can be, you can say, transferred into E. coli or any other host cell. Clear? So, for example, the human gene, okay, it was isolated from humans. Okay, this gene was inserted in a plasmid, which is acting as a vector, and this vector was then transferred into the E. coli. Okay, then E. coli start producing the, you can say, the product which was coded by the human gene. Okay, so insulin is being produced. Human insulin is being produced in bacteria and yeast. Okay, now which bacteria is usually used? E. coli is usually used as a host cell or host organism, because E. coli can divide very fast. It is very easy to culture these bacteria. Okay, they don't require any special medium. They can grow in minimum medium. Okay, they don't require much space to cultivate them. Okay, you can produce the desirable product in large quantities. Okay, and obviously they multiply rapidly. This is also used in biotechnology. Okay, so these were the plasmid. So you can see this is an image showing you plasmid. This is the bacterial genomic DNA. Okay, and then you can see these are the plasmid. Okay, this is the, you can see these are circular, they are double stranded, they are small. Okay, clear? And they also consist of some selectable marker genes such as antibiotic resistance. Now let's move on. Now let's talk about cell envelope and its modifications. The most prokaryotic cell have a chemically complex cell envelope. Okay. The envelope consists of 3 tightly bound layers. Now, what are these 3 layers? Okay. Outermost layer is known as the glycocalyx. Then the middle layer is known as the cell wall, and the innermost layer is known as the plasma membrane. Okay, so first we will talk about glycocalyx. So glycocalyx is the outermost layer, okay, of the cell. And what is the function of glycocalyx? First of all, first function is the protection. So it protects the cell from the harsh environmental conditions. Clear? So this is the first role of glycocalyx. Second role of glycocalyx is adhesion. So glycocalyx help the cell to adhere to the surfaces. Okay, so these are the 2 very important functions of the glycocalyx. So glycocalyx, they are found in bacteria and other prokaryotic cell as well. And glycocalyx, this is a viscous gel-like substance. Clear? And what is the composition of glycocalyx? It is made up of polysaccharides. Or it can be also made up of polypeptides as well. Clear? So this was about glycocalyx. Okay. We have discussed about the function of the glycocalyx as well. So here you can see, this is an image showing you the capsule in the cell wall. So glycocalyx Sometimes glycocalyx is also referred to as capsule. But when the glycocalyx become very thick, then it is known as the capsule. Okay, so we'll talk about this capsule also and the slime layer also. Okay, so you can see in this image, you can see the capsule, which is basically glycocalyx when it is thick. Okay, you can see the cell wall here, which is the middle layer, and then the plasma membrane, which is the innermost layer. So this is a image showing you. Okay, this is a bacteria, this is the prokaryotic cell, and the cell envelope is shown to you. So cell envelope consists of 3 tightly bound layers. Okay, let's talk about the glycocalyx first. So it varies in composition and thickness. So it can be made up of polysaccharides or polypeptides as well. Okay, now if the glycocalyx is in the form of loose sheath, then it is called slime layer. Okay, but if glycocalyx is thick and when it is tough, it is known as capsule. Okay, so in the previous image, I've shown you capsule. Okay, so glycocalyx, this is called capsule when the glycocalyx become thick and tough, and this protects the cell from harsh environment. Okay, glycocalyx also help in the adhesion of the prokaryotic cell to the host cell. So here you can see this is image showing you glycocalyx, slime layer, and capsule. So this is the you can say slime layer, okay, which is in the form of loose sheath, and then you can see this is the capsule when the glycocalyx is very thick and tough. Clear? Now let's move on. Let's talk about the cell wall now. The key components and features of the cell wall. So basically, the cell wall in bacteria, it is made up of peptidoglycan. Okay, cell wall of basically eubacteria. Eubacteria also known as the true bacteria. So cell wall of, you can say true bacteria actually, it is made up of peptidoglycan. Cell wall of archaebacteria, okay, they have different composition. Okay, so instead of peptidoglycan, they have some other polysaccharides. Okay, they have some other proteins and glycoproteins. Okay, so here the bacteria, we're talking about true bacteria here. Cell wall is made up of peptidoglycan. Peptidoglycan is also known as murine. Okay, archaebacteria consists of pseudomurine. This peptidoglycan, this is a mesh-like polymer. Clear? So this peptidoglycan consists of alternating N-acetylglucosamine, which is known as NAG, and N-acetylglutamic acid molecules. So remember, this is very important. Okay. So you can see this peptidoglycan is made up of NAG and NAM, which is alternating with each other. Okay, you have NAG, then NAM. You have NAG, then NAM. Okay, and this forms the peptidoglycan. Okay, so this is peptidoglycan is a unique polymer which is found in cell wall of most of the bacteria. And what is the function of this peptidoglycan? So this peptidoglycan provide structural support and help the cell withstand internal osmotic pressure. Clear? Peptidoglycan, as we have discussed, is a mesh-like structure which is made up of this NAG and NAM. Clear? Okay, now this NAG and NAM, they are cross-linked by peptide bridges. Here you can see in this structure, this is showing you cell wall and plasma membrane of the prokaryotes. Cell wall, we have discussed, it is made up of peptidoglycan. So this is the cell wall you can see. Okay. Below the cell wall, you have the plasma membrane. This is the plasma membrane. This part is the cell wall. And you can see this cell wall is made up of peptidoglycan. And between this cell wall and the plasma membrane, you can see there is a periplasmic space. This is periplasmic space. Okay. So this periplasmic space divides the cell wall and the plasma membrane. You can see here the peptidoglycan, it is made up of NAG and NAM, NAG and NAM, NAG and NAM. So they are alternating with each other and they are joined together with the help of peptide bridges. Okay. Now let's move on. Clear? Now let's talk about the function of the cell wall. So first of all, what is the first function? It protects against the osmotic lysis. Now what do you mean by that cell wall protects against osmotic lysis? So lysis means breakdown. Now it means that the cell wall prevents the cell from bursting when water enters into the cell due to osmosis. And how does this protect the cell? By providing mechanical strength to the cell. So this cell wall provides mechanical strength to the cell and prevents the bursting or lysis of the cell when the water enters due to osmosis. So this is known as osmotic lysis, breakdown of the cell or lysis of the cell when the water enters into the cell due to osmosis. Okay, we all know about osmosis and diffusion. Clear? Osmosis is the movement of solvent. Okay, in diffusion there is a movement of solutes. So there is a difference between diffusion and osmosis. Okay, movement of water or movement of solvent from low concentration to higher concentration, this is known as higher concentration of solute, this is known as osmosis. Second function is maintaining of the cell shape. So cell wall provides rigid structural support to maintain the characteristic shape of plant, bacterial and fungal cell. You can see in which organism cell wall is present. So cell wall is present in bacteria. Okay, it is present in some protists also. It is present in fungi. In fungi, cell wall is made up of chitin. Then plants also have cell wall. In plant, the cell wall is made up of cellulose. In bacteria, the cell wall is made up of peptidoglycan. Now, animal cells, they lack the cell wall. Instead of cell wall, they have the cell membrane. Okay, next function is act as a barrier against harmful substances. So it forms a protective layer that blocks the entry of the pathogens, toxins and other damaging materials. So it is acting like a barrier, okay, for the entry of the pathogens. Okay, toxins and other damaging materials as well. Now next one, helps in the cell recognition. Now what do you mean by helps in cell recognition? So this cell wall contains specific molecules that allow cells to identify and communicate with the neighboring cells. So this communication between the neighboring cell is very important. Okay. So here you can see this image we have discussed. This is the image showing you cell wall in the plasma membrane of the prokaryotes. So below you can see this is the innermost layer, plasma membrane. Then middle layer is the cell wall, which is made up of peptidoglycan. Okay. Now let's talk about the classification based on cell wall composition. So bacteria, they can be gram positive or they can be gram negative. Okay, what do you mean by gram positive bacteria? So gram positive bacteria, they will retain the purple stain. Yeah. So bacteria, they stain purple due to the dye known as crystal violet dye. So this stains the bacteria purple. Okay, now what is the structure of gram positive bacteria? So they have a thick peptidoglycan layer, okay, which is 20 to 80 nanometer. Now, next one, they have single lipid membrane as well, means lipid content is less. They have teichoic acid present, okay, in the cell wall, and then they appear purple or violet after gram staining. So, in gram staining, what you do? Obviously, this gram staining was discovered by Gram, okay, and then, okay, why do they appear purple or violet? So, these bacteria, when they are stained with crystal violet, they retain this dye. That's why they appear purple or violet. Okay. So you can see the peptidoglycan layer is this is quite thick. Okay. So because this is thick, so it helps in retaining of the crystal violet dye. And also the amount of lipid is also less. So what happens? You stain with the crystal violet dye and then you decolorize it using the alcohol or acetone. Okay. And then you again restain it with safranin. So in gram-positive bacteria, as there is a thick peptidoglycan layer and the lipid content is less, so there is no effect of decolorizing agent, which is alcohol. Okay, alcohol or acetone can be used as a decolorizing agent. Okay, so there is no decolorization, and when the safranin is added, so the you can say these bacteria they don't take up the safranin because they are already stained with the crystal violet dye, so they will appear purple or violet in color. Okay, examples of gram positive bacteria, you should remember these examples. This is very important. Okay, first is Staphylococcus, second is Streptococcus, third is Bacillus clostridium and Listeria. These are all gram positive bacteria. Okay, so here you can see this is cell wall of the plasma membrane of the prokaryotes. So in the case of gram positive bacteria, the peptidoglycan layer is quite thick. Okay, and because of this, it can retain the crystal violet stain. Next, another category is gram negative bacteria. Now, how gram negative bacteria is different from gram positive bacteria? So let's talk about the structure of gram negative bacteria. So they have a thin peptidoglycan layer, which is 2 to 3 nanometer. They have double lipid membrane, inner and outer. Okay. So because of this, the gram negative bacteria, as the lipid content is more, okay, the peptidoglycan layer is surrounded by an outer lipid membrane. Okay. So they are double lipid membrane. So the alcohol or the decolorizing agent can easily decolorize these bacteria. So this crystal wall stain is removed. Why? Because it has a thin peptidoglycan layer and it has a double lipid membrane. Okay. So when you expose these cells to alcohol or acetone, then the stain is removed. And then when you again stain it with safranin, they take up safranin dye and they appear as pink color. So gram negative bacteria will appear pink in color. Okay. So through gram staining, you can determine whether the bacteria is gram positive or gram negative. Okay, so you can see the gram negative bacteria, they contain lipopolysaccharides. They will appear pink or red after gram staining because the crystal violet dye, this is, you can say, removed by addition of the alcohol. Examples are Escherichia coli, Salmonella, Pseudomonas Neisseria, and Haemophilus. These are the example of gram negative bacteria. You can see this image showing you the cell wall of gram negative bacteria. Okay, so here you can see this is the plasma membrane. Okay, then here you can see this is the cell wall above the plasma membrane. Cell wall is made up of a thin peptidoglycan layer, and above the peptidoglycan layer there is a this layer, lipid layer. Okay, here you can see Here they have labeled NAG and NAM. The peptidoglycan, this is made up of NAG and NAM, which are alternating with each other. Okay, so because of the presence of a double lipid membrane outside the thin peptidoglycan, the stain is not retained after adding the decolorizing agent. And when you add the safranin, then these cells, these bacteria, they take up the safranin dye and they appear as pink or red. Next, let's talk about the plasma membrane. So the cell membrane of the prokaryotes are also known as the plasma membrane. Okay, prokaryotic cell membrane is a phospholipid bilayer. Okay. Now the plasma membrane is composed of lipid and proteins following the fluid mosaic model. Okay, so here also the plasma membrane of the prokaryotic cell, this is a phospholipid bilayer, means two layers. Okay, it is typically 7 to 8 nanometers thick and consists of phospholipids. Okay, so these are molecules with hydrophilic heads and hydrophobic tails. The phospholipids, they have hydrophilic heads, means water loving heads, and hydrophobic tails, means water repelling tails. So tails are water repelling, heads are water, you can say philic or water loving. Remember this, heads are water loving, tails are water repelling. Okay, so this plasma membrane also consists of proteins as well. Okay. So proteins are integral proteins, means they are spanning the entire membrane. Okay, and there are peripheral proteins also which are attached to the surface. So proteins are of two types, integral proteins and peripheral proteins. So here you can see this is an image showing you plasma membrane of the prokaryote. Clear? So here you can see the plasma membrane consists of these peripheral proteins. Okay, it consists of glycoproteins, globular proteins, glycolipid as well, then cholesterol, carbohydrate. So these, you can say this is the image showing you plasma membrane of the prokaryotes. And you can see here, this is a lipid bilayer. And you can see here, the tails and header also can be seen here. Okay, these are phospholipids. Okay, which have a tail and a head. Okay, we were talking about the plasma membrane. What are the key function of the plasma membrane? First is selective permeability. Okay, means they allow specific molecules to pass through them. This is known as selectable permeability. So the plasma membrane controls which substance can enter or exit the cell. So it is acting as a selectable filter. Okay, so this is the first key function of the plasma membrane. Now next one, transport of nutrients and waste. So this plasma membrane facilitates the movement of essential nutrients into the cell, and it will remove the waste product, okay, from the cell. So the cell, it is carrying out the metabolic activities. It is a living cell. Living cell will be metabolically active. A lot of reactions are going on, biochemical reactions, and the waste product will be produced. So nutrients are absorbed and the waste material is expelled out. The next one is energy production. So how is plasma membrane involved in energy production? So it contains proteins involved in electron transport chain and ATP synthesis. Here. Next one is cell signaling. So the plasma membrane contains the receptor protein that receives and transmits the signal from the environment as well as the other cells. Okay, it helps in maintaining the cell shape. Plasma membrane, it provides structural support and helps maintain the cell's specific shape through membrane proteins and cytoskeleton attachments. Clear? Next one is protection from the external environment. Clear? So this plasma membrane also provides protection to the cell from the harsh external environment. Here you can see this is an image showing you plasma membrane of the prokaryotes. Here again you can see these are the phospholipid. Integral proteins are present. Okay, channel proteins are present. This is an integral protein. Then you have the peripheral proteins present on the periphery. You have the channel proteins here. Then you can see you also have glycolipids here. Cholesterol you can see. Glycoproteins are there. Glycolipids are there. Carbohydrates are there. Globular proteins are there. Okay, these are the components of the plasma membrane. Okay, now let's talk about the membrane extensions. First one is the mesosome. So mesosomes are specialized differentiated form of cell membrane formed by extension into the cell. Okay, now what are the function of mesosomes? First one is cell wall formation. Second is DNA replication. This is a very important function. Next one is distribution. You can say to the daughter cells. And respiration. Respiration is also very important function of mesosome. And secretion processes as well. So these are the two major functions, respiration and DNA replication. Okay. Chromatophores. These are membranous extensions into the cytoplasm in some prokaryotes like cyanobacteria which contain pigments. So cytochromes, chromatophores are present in cyanobacteria. Clear? The question can come from here. Chromatophores consist of pigments. Here you can see this is an image showing you a bacterial mesosome. So mesosomes are generally involved in respiration. Okay. Yeah, so you can see this is an image of the mesosome. It has a mesosome sac. You can see these are the plasma membrane. This is the cell wall. Clear? And mesosomes, they are formed by the extensions. Okay. Now motility structures which help in locomotion. Okay, structure which helps, which gives motility to the cell. First one is flagella. Flagella filamentous extensions from the cell wall for motility. Okay, the flagella is composed of filament, hook, and basal bodies. These are the three parts of the flagella. Remember this. Pili. What are pili? So they are elongated tubular structures made of a special protein for conjugation. So conjugation is a type of sexual reproduction where two bacteria, they come close to each other. They form this sex pili. And through this pili, DNA can be transferred. Okay. Pili also helps in the attachment of this bacteria to the host tissue as well. Fimbriae. Fimbriae are small bristle-like fibers sprouting out of the cell for the attachment. So fimbriae, what is the function of fimbriae? Attachment. It helps in the attachment of the, you can say, the bacteria, or it helps in the attachment of the prokaryotic cells to the host surface. Clear? So this is fimbriae. Now next one. Clear? So fimbriae are basically, they help in the attachment of these prokaryotes onto the rocks as well. Okay, or on the host tissues. This is an image showing you flagella, pili, and the fimbriae. Where you can see these are the pili, which helps in the conjugation process as well. This is the fimbriae, which helps in the attachment of the bacteria to the rocks in the streams as well as to the host tissue as well. These are the flagella, which provides locomotion, which provides motility to the, you can say, bacterial cell or prokaryotic cell. Now next one is ribosomes and inclusion bodies. So first one is ribosomes. So these are associated with the plasma membrane. They are the site of protein synthesis. This is very important. Made up of two subunits, 50S and 30S, which together form the 70S prokaryotic. So in prokaryotes, 70S ribosomes are present. Now, polysomes, also known as polyribosomes, this can be seen in prokaryotes. Okay, what do you mean by polyribosomes? Poly means many, so several ribosomes may attach to a single mRNA. Okay, and then they form a chain. So when several ribosomes attach to a single mRNA, what happens then? Multiple copies of a single protein is made. So because of the polysome, what happens? Multiple copies of a single protein is made. This is the image showing you polysomes. Here multiple ribosomes are attached to the mRNA. Clear? Let's talk about inclusion bodies. So these are the reserve material are stored in the cytoplasm. These are known as the inclusion bodies. They are basically reserve material which are stored in the cytoplasm. They are not bounded by any membrane system. Examples are phosphate granules, cyanophycin granules. So cyanophycin granules, they are present in cyanobacteria. Okay, and they store cyanophycin starch, which is very similar to glycogen. Okay, and there are glycogen granules also. Gas vacuoles are also present. These are found in blue-green, okay, and purple and green photosynthetic bacteria. What is the function of gas vacuole? They help with buoyancy. They provide buoyancy to these bacteria. This is showing you gas vacuoles in blue-green algae. Okay, these provide buoyancy to these bacteria because they are aquatic. Okay, now let's summarize this entire lecture. We talked about the cell envelope. Okay, it consists of cell wall, cell membrane, and capsule. Okay, capsule, we call glycocalyx as capsule. When the glycocalyx is thick and tough, then we call glycocalyx as capsule. Otherwise, we call it as slime layer. Okay, cell wall is made up of peptidoglycan. Okay, and cell wall provides structural support. Cell membrane regulates transport and contains enzyme as well. Cell membrane is also known as plasma membrane. Okay, so gram-negative bacteria, they have a thin peptidoglycan, and towards the outside of this thin peptidoglycan membrane, a lipid layer is also present. Modification of cell envelope. First is capsule, which provides protection and also helps in the attachment of the bacteria to the host tissue. Then you have the pili, okay, as well as fimbriae. Both of these help in the adhesion of the bacteria to the host tissue. Then you have the flagella for movement. These are the modification of the cell envelope. Okay, we talked about the ribosomes also. So in eukaryotes, you have 70S ribosomes. So there are two subunits. Okay, smaller subunit 30S, larger subunit 50S. It is a site of protein synthesis. They are found free in the cytoplasm. Inclusion bodies, these are the storage granules for nutrients, contain metabolic reserves. Example is glycogen, lipids, and phosphate granules. These are known as inclusion bodies. Now there are some questions related to these topics. So first question is, which of the following statement about bacterial glycocalyx is incorrect? Means you have to tell about the false statement. The first option is, it consists of polysaccharides and proteins secreted by bacterial cell. Okay, next is, it helps bacteria adhere to surfaces and biofilms. Third option is, the glycocalyx is always thick and visible under light microscope in all bacteria. Fourth is, it can protect bacteria from dehydration and host immune responses. What will be the correct answer? Let's have a look at the answer. Okay, so option number C is the correct answer. Glycocalyx is always thick and visible under light microscope in all bacteria. Okay, so this is false because glycocalyx can vary significantly thickness and structure among different bacterial species. Some bacteria produce thin, loosely attached glycocalyx known as the slime layer. Others produce thick and well-organized glycocalyx called as capsules. In many bacteria can exist without a visible glycocalyx as well. Now, the thickness and presence of glycocalyx often depends on the environmental conditions. Now, next question, which of the following statement about the plasma membrane's phospholipid bilayer is incorrect? Means which is false. Options are, option number A, the hydrophilic heads face the extracellular fluid and cytoplasm. Option number B, hydrophobic tails face each other in the middle of the bilayer. Option number C, hydrophilic heads are water soluble. And option number D, hydrophilic tails are oriented towards the aqueous environment. What will be the correct answer? So here they are talking about this head and the tail of the phospholipid. Okay. So which is hydrophilic, which is hydrophobic? Let's have a look at the answer. The correct answer is option number D. The hydrophobic tails are oriented towards the aqueous environment. Okay, so hydrophilic means water loving. Okay, so the hydrophilic head face the aqueous environment, both inside the cell, which is cytoplasm, and outside the cell, which is extracellular fluid. Clear? The hydrophobic tails face each other in the middle of the bilayer, away from the water. And this arrangement is energetically favorable and creates a stable membrane structure. This organization is crucial for maintaining the selective permeability of the cell membrane. Okay, so this cell membrane also known as the plasma membrane. Okay, this provides protection also. Okay, cell wall also provide protections against the pathogens as well. Okay, so this is selectively permeable plasma membrane. Okay, so it will allow entry of only certain, you can say compounds or chemicals, okay, or any solute to enter. It will not allow other particles to enter into the cell. Some keynotes for you. Cell envelope consists of 3 layers: cell wall, cell membrane, and capsule. Some bacteria have pili for attachment and conjugation as well. So pili help in conjugation as well. Pleasure enable movement. 70S ribosomes synthesize proteins. In prokaryotes, inclusion bodies store nutrients. Gas vesicles or vacuoles help in buoyancy. Carboxysomes aid in carbon fixation. Some interesting questions for you. How do teichoic acid contribute to bacterial pathogenicity? Teichoic acids, okay, they are present in the cell wall. Okay, you have to think about this. What role do peptidoglycans play in bacterial cell wall rigidity? Okay, you have to explain the role of peptidoglycans. Then next one, why are 70S ribosomes unique to prokaryotes only? Because prokaryotes, they have a TS ribosome. Next one is, how do inclusion bodies help bacteria survive in extreme conditions? Next is, what is the significance of capsule in bacterial virulence? Okay, virulence means they can cause disease. So think about these questions. Okay, now we have discussed the theory. We will now discuss the DPP related to these topics. Okay. Okay, let's start with the first question. So, which of the following is not a characteristic feature of prokaryotic cell? First is presence of mesosome. Second is circular DNA in cytoplasm. Third is 70S ribosome. And fourth is well-defined nucleus. What will be the correct answer? Okay, they are saying which is not a characteristic feature of prokaryotic cell. Okay, so well-defined nucleus will be the correct answer. Okay, they are asking not a characteristic of prokaryotic cell. The prokaryotic cell doesn't consist of a well-defined nucleus. Instead of nucleus, they have nucleoid. The next one, the cell envelope in some bacteria have an outer layer called glycocalyx. Which of the following is a thick and tough glycocalyx? Slime layer, capsule, mesosomes, or fimbriae. So when the glycocalyx is thick and tough, what is it called? Is it called slime layer, capsule, mesosomes, or fimbriae? What would be the correct answer? Okay, it is called capsule. When the glycocalyx is thick and tough, it is called capsule. When it is thin, it is known as slime mold. Next one, which structure in prokaryote is responsible for increasing the surface area of plasma membrane and aiding in respiration? Okay. Which structure will help in increasing the or enhancing the surface area of the plasma membrane and also helps in the respiration? So what would be the correct answer? The answer will be mesosomes. Option number B is the correct answer. Next one, what is the function of bacterial pili? Motility, attachment to host tissue, protein synthesis, or storage of reserve materials. Okay, option number B will be the correct answer, attachment to the host tissue. Okay, so option number B is the correct answer here. Here, this pili also helps in conjugation also. It also helps in the attachment of the bacteria to the host tissue as well. Next, inclusion bodies in prokaryotic cells are associated with DNA replication, storage of reserve materials, photosynthesis, or formation of cell envelope would be the correct answer. Okay, option number B will be the correct answer here. Storage of reserve materials. Okay, inclusion bodies. Okay, they are associated with storage of the reserve materials like glycogen bodies. Okay, here cyanofacial granules, phosphate granules. Clear? So we have discussed the DBP also. Okay. So now we will end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.